Dear students, today we are going to discuss the concept of genome and the concept of genomics. Genome is the complete set of DNA of an organism, whereas genomics is the study of the genome of an organism, its entire genetic material in the form of RNA, DNA, and chromosomes. So, genome is simply the complete set of DNA, whereas the study of the genome is genomics. But before moving on to the genome, we should be having an idea that what cell is, where the nucleus is, where the chromosomes are, where the DNA is present, where the nucleotides are present, and then how these nucleotides transcript and this translate with the help of the genetic code for the formation of the protein so that a complete information is decoded. So when we study all these things, actually we are studying a genome. So before moving on, we should be having an idea rightly from the cell up to the formation of the proteins. As we all know that the cell is the structural and the functional unit of all the living organisms. It is the smallest unit of life. Some of the organisms have only one cell. We call them as the unicellular organisms. Or they have many cells. They are made up of many cells. We call them as the multicellular organisms. Approximately an organism such as this human being, it is made up of about 40 trillion cells. Even the only the brain of the human beings is made up of 80 billion cells. Now, when we talk about a cell, it is a complete entity which is capable, which is able to carry out all its function in a single cell. That is, it means it has all the required things for the survival. But we should be knowing that a cell, a cell which has besides other organelles, I shall be showing you, besides all other organelles, it has one region called as nucleus, wherein the chromosomes are present, wherein these chromosomes are made up of DNA, and this very DNA, we call it the substance, which is composed of two polynucleotide chains that coil around each other to form a helix. Now, this DNA is called as the deoxyribose nucleic acid, and it is a substance which is called as the hereditary material. It is made up of same chemical and physical components in all the organisms. The DNA sequence is particular side by side. In terms of the arrangement of bases along the DNA, that is the bases adenine, guanine, cytosine, and uracil, or thymine. This order, the order in which these bases are present, it distinguishes an organism from the other organism. It is particularly the arrangement of these bases which distinguishes a human being from a cat, a human being from a cow, a human being from a rat, or it is able to differentiate in between the cat and the rat. It is just only the arrangement of the nucleotide base pairs which differentiate different kinds of the organisms. Now, when we talk about the DNA, it is called as the genetic material because it is the material which carries information from the parents to the offsprings. It 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 is the carrier of the information or it is the control room which controls the instruction for the development, functioning, growth, reproduction of all the organisms, including human beings. DNA strands, such as these very polynucleotides, are composed of single monomeric units called as nucleotides. As I told you, that it is cytosine, guanine, adenine, thymine, or adenine, guanine, cytosine, and thymine. A sugar called as a deoxyribose and phosphate group. These are joined by the covalent bonds. We call it as the phosphodiester linkage bonds. This bond joins the sugar of the one nucleotide to the phosphate group of the another nucleotide, thus forming a backbone. These both strands of DNA store the biological information. The information is replicated and when two as and when these two strands separate. For example, this very strand is separating here. The information is replicated from one into two different strands. The large part of this very large part of this very DNA strand, it is a non-coding strand in all organisms. It means almost approximately in the case of human beings, 98% of the DNA is non-coding, which we called as the junk DNA. That it has no role in the passing on the genetic information from the parents to the offsprings. DNA is organized into long structures called as the chromosomes. The chromosomes are duplicated in the process of the DNA replication, providing the complete set of chromosomes for the daughter cell. The DNA was first isolated by the Friedrich Miescher in 1889, and the structure was identified, as we all know, the structure was identified by the Watson and Crick. The sequence of this very nucleotides, DNA or RNA, it encodes for the synthesis of a gene product. It can be either RNA or protein. As we all know that this very information is passed on in the form of a genetic code, that is adenine, guanine, cytosine, and uracil or thymine placed in a particular sequence. This forms a genetic code which is a triplet code, and we all know that by the process of the transcription, translation, and then the formation of the protein. Now, as we all know that this very genome. Now, the total amount of the DNA content in one copy of a single complete genome, it comprises the genome size. Now, as we got to know that the genome is the complete set of genes in an organism. Now, there are certain interesting facts about the genome. Virus, one of the smallest genome is of the virus bacteriophage. It is 3569 base pairs. Then bacteria, that is Escherichia coli, it is 4 into 10 raised to power 6 base pairs. The first multicellular animal genome was decoded, was that of the nematode Caenorhabditis elegans. It is 9.8 into 10 raised to power 7 base pairs. The smallest vertebrate genome is of a fish, that is Tetrodon. You must have heard about the name Tetrodon. It is commonly known as the puffer fish. It has the genome size of 3.85 into 10 raised to power 8 base pairs. And we all know Homo sapiens, that is the main. It has the genome size of 3 into 10 raised to power 9 base pairs, which is around 3 billion base pairs. The largest vertebrate genome is again that of the fish. We call that very fish as the Protopterus Ethiopicus aka. It is a marbled lungfish. It has a genome size of 1.3 into 10 to the power 11 base pairs, the longest genome size, even more than that of the human beings as well. So the complete information or complete set of the genes, it is called as the genome. Now we got to know that the cell contains the nucleus contains the chromosome contains the DNA. It is the DNA which has the bases in the form of adenine, guanine, cytosine, and uracil, which are present in a particular sequence. And this very sequence is decoded in the form of the genetic code to form the protein by the process of the transcription and translation. And this amount of the size of the genome or the size of the genes, complete size of the genes are the total amount of DNA contained in one copy of the single complete genome. We call this as the genome size. So simply the genome is the complete set of genomes. But when we try to differentiate it, try to explain it in the form of genome size, it is the total amount of DNA contained in the one copy of a single complete genome. Next, as we have discussed earlier, the DNA in the human genome is arranged in 24 distinct chromosomes. It, as we have discussed in our earlier slide, that 22 are autosomes, that is 22 pair are autosomes, and one X and one Y chromosome. They physically separate molecule that range in length of about 50 million to 250 million base pairs. Each chromosome contains many genes, the basic, physical, and the functional units of the heredity. Genes are specific sequences of bases that encode instructions on how to make proteins. So genes comprise only about 2% of the human genome. As I told you earlier, that 98% of the DNA, it is called as junk DNA. It has nothing to do with the passing of the instructions or passing of the information or passing of the hereditary characters. The remainder consists of the non-coding regions. 2% of the human genome comprises the gene comprises only about 2% of the human genome, whereas remainder is the non-coding region, so whose function is only to provide chromosomal and structural integrity and regulating where and when in what quantity the proteins are made. So they are only the sporting you can say sporting structures, sporting components of only 2% of the genome. A human genome is estimated to contain about 20,000 to 25,000 genes. Let us talk about the term genome and genomics. The term genome, as we all know, gene and chromosome. It is the culmination of the gene and chromosome, G from the gene and O from the chromosome. So it is genome. In 1920, Hans Winkler, a professor of botany in Hamburg University, Germany, he coined the term this very genome by the combination of gene and chromosome in 1920, whereas the genomics, the component which arose from the genome, was coined by the term Roderick. Tom Roderick. He was genetist in the Jackson Laboratory in 1986, almost 66 years after the coining of the term genome. The genomics term was coined in year 1986. Now, in the earlier part, we discussed a little bit about the genome, as we discussed that it is the sum total of the genetic information of an organism which store biological information. It can either be DNA or RNA. So DNA or RNA capable of storing all the information in a complete set, we call that very part as a genome. So all the eukaryotes and the prokaryotes have DNA genome, but viruses, as we all know, that they have either DNA or RNA as their genome. As we all know, that there are certain viruses which are double-stranded, which have the double-stranded DNA. Some have single-stranded DNA. Some have RNA as their DNA, RNA as their hereditary material. So there is there are different you can say kinds of the genome present in different organisms. So, to elaborate the genome, we say the complete set of DNA, including all its genes. So, each genome contains all the information needed to build and maintain that organism. So, in humans, as we talked earlier, there are about 3 billion DNA base pairs which are contained in all the cells that have a nucleus. And it refers to a total amount of the DNA of a single cell haploid in the case of diploid organisms of an organism including its genes. So when we say complete set of the genes, it is haploid cell in the case of diploid organism. So in a diploid organism, we thou are having the um, 23 from the mother and 23 chromosomes from the father. So 23 are enough. So it is the complete information. It is the complete information present in an organism. So it comprises the genome. So genomes also vary widely, as I told you earlier, that it ranges from the 3569 only 3569 base pairs in the case of virus bacteriophage to about 1.3 into 10 raised per 11 base pairs in the case of Protopterus aethiopicus aka. So there is a wide, you can say, range of the genome size. The smallest known genome for a living organism, bacterium, contains about, you can say, 6 lakh DNA base pairs, while human and the most genomes have about 3 billion base pairs. So the red blood cells don't have the, you can say, they do not have the nucleus. That is why they don't have a complete, except the red blood cells, because they don't have nucleus, so they don't have the genome. So genome types, when we talk about the types of the genome, we have the viral genome, we have the bacterial genome, we have the eukaryotic genome. As we have talked earlier, that there are viral genomes which are composed of either RNA or DNA. The genomes of the RNA viruses can be either single-stranded or double-stranded RNA. They may contain one or more separate RNA molecule segments, or some DNA viruses can have either single-stranded or double-stranded DNA as well. The prokaryotes and eukaryotes have DNA genomes. Archaea have the single circular chromosome. Most bacteria have single circular chromosome. However, some bacterial species have linear chromosomes or the multiple chromosomes. And in the case of eukaryotic genome, we know that they are composed of one or more linear DNA chromosomes, as we have discussed earlier in our introductory, you can say, opening of the lecture, that they are composed of the DNA chromosomes. The number of chromosomes vary widely from jack jumper ants. In the case of jack jumper ants, and an asexual nematode, each have only one pair, to a fern species that has 70 pairs of the unique, you can say, chromosomes. So this is a little bit of a depiction of this very coiled RNA in the case of bacteriophage, and this is Escherichia coli DNA. It is circular. Here in this very case, it is the RNA, which is a coiled RNA as the genetic material. Here it is a circular genetic material in the case of bacteria.